Next, we'll go over Mendelian as well as some more complicated genetics and Punnett squares. We can also take a look at more complicated genetics. The traits that we've looked at and Mendel looked at were very, very straightforward, meaning that you had a dominant allele or a recessive allele, and the dominant would completely mask that recessive allele. However, this is not always the case. You can have what is termed incomplete dominance. This is when you don't have one trait being completely dominant over the recessive state. As a result, your heterozygous individuals share a phenotype that is in between the two pure breeding plants. This is very characteristic when you see a blending of two traits. An example is if you're crossing red flowers with white flowers, red being dominant, white being recessive. The heterozygous individuals that would result from that would have pink flowers, the blending of the red and white traits. You can also have what is termed codominance. In this instance, two alleles are both fully expressed, meaning that they will be codominant to each other. This is because one allele isn't completely dominant over the other, and the other one isn't completely recessive. So as a result, when you have both alleles present, both of them are going to be expressed. A good example of this is your ABO blood typing. When you're considering blood types, there are three possible alleles that you can have: the dominant A, the dominant B, and the recessive state, which would be for the blood type O. If you have an allele that has the A antigen, or IA, you would be expressing the A antigen on your blood cells, which means that your blood cells would have the cell surface marker consistent with blood type A. If you have IB, this means that you would be expressing the B antigen on your blood cells. However, I is considered to be recessive, and if you have the recessive alleles, you do not express anything. Keep in mind that with blood types, you have, as an individual, room for two alleles. So this means that if you have blood type A, you could have blood type A and have two different genotypes. Either you have IA or you have IA with little I, meaning that you have a recessive allele but the dominant A allele as well. In both of those instances, you would be expressing blood type A. Similarly, if you had blood type B, you could be homozygous dominant, meaning that you would have IB, or you could be heterozygous, meaning you have IB little I. However, if you have blood type AB, this is the codominant state, meaning that you have IA and IB and you express both antigens on your cell. There's only one way to have blood type AB. Your genotype would have to be IA-IB. This slide here depicts the concept of codominance versus incomplete dominance. Whenever you see both parental traits being exhibited in your offspring, that is indicative of codominance, which we can see in the flowers that are both white and pink at the same time. We can also see that in the snake, that we have both skin patterns of both parents are expressed in that one individual. This is an example of codominance. However, when you have incomplete dominance, you get the blending of the two traits, which can be seen in the pink flower. This diagram here illustrates the concept of incomplete dominance. We have a red snapdragon crossed with a white snapdragon. The only gametes that can be produced by the red individual are ones with C and a big R. The only gametes that can be produced by the white individual would be CW. The F1 generation for this specific cross would be all hybrid individuals. Once again, with a hybrid individual, half of those gametes would be CR and the other half would be CW. If we want to get our F2 generation, we would cross those F1 individuals together or self-pollinate them. We can see what the resultant F2 generation would be. Once again, with two heterozygous individuals, we would have eggs being produced from one, which would have half of them would have CR, the other half would have CW, and similarly, the other parent producing the sperm would also have similar gametes, half of them being CR and half being CW. When you carry out that Punnett square to figure out what your offspring ratios would be, one individual would be CR-CR, obtaining the CR allele from the egg and another CR allele from the sperm. And two of those offsprings would have CR-CW and would be heterozygous, and then one individual would be inheriting the CW from the egg and the CW from the sperm. If we take a look at the phenotype ratios, we can see that concept of blending in the heterozygous individual, which indicates we're dealing with incomplete dominance. So our F2 generation would be one red individual, two pink individuals, and one white individual. This slide depicts the concept of blood typing. So as stated in the previous slide, we have three different alleles for blood types, IA, IB, and I. These alleles are going to be expressed by the different antigens that you would be carrying on your red blood cells. So let's take a look at the different genotypes that we could have. If you have IA, or if you have IA small I, you would exhibit the blood type A. And this would be exhibited by the cell surface marker carbohydrates that you have and carry on your red blood cells. Similarly, if you had blood type B, that would be from the genotype IBIB or IB little I. And you can see below that you would have different cell surface markers consistent with blood type B. If you have IAIB as your genotype, you would exhibit both of those cell surface markers, both the A antigens as well as the B antigens. And then finally, if you have the fully recessive state, you have no surface carbohydrate markers, and hence you would be considered blood type O. There are also important concepts to understand when dealing with the extension of our simple Mendelian genetics. You can have pleiotropic genes, and this occurs when one gene has multiple phenotype effects. You can also have polygenic genes when one trait is controlled by multiple genes. This is very consistent for traits that exist on a scale, such as skin color, height, and weight. We can also have epistasis occur, and this is when one gene controls the expression of another. A common example of this is dealing with albinism. You can carry a gene for hair color pigment. However, if you carry both recessive alleles for albinism, then you lack the ability to make pigment. So it doesn't make a difference what you have for your pigment producing alleles because you don't have the ability to make the pigment anyway. This is a classic example of epistasis. Here we've got an example of epistasis. Let's say we're doing a dihybrid cross and we're dealing with two heterozygous individuals, meaning that we've got big C, big B, little b for our hamster or guinea pig alleles. We are also crossing this male with a female of a similar genotype. So we've seen before that we need to predict what our gametes would be from those individuals. So if I have big C, big B, little b, I have four potential gametes that could result, according to the law of segregation and law of independent assortment. I could have a big C, big B gamete. I could have a little c, big B gamete. I could have a little c, little b gamete, as well as a big C, little b gamete. And this is what we see in the gamete possibilities, which are listed at the top of the diagram as well as on the side. When we go ahead and cross those gametes and simulate fertilization, we get our potential offspring, which are all depicted in those squares below. Note that we have the genotype as well as the phenotype. So when we're dealing with albinism in guinea pigs, if an individual has little c, they are going to be an albino. And this is what we see here. So it doesn't matter whether or not you have big B as your other alleles, big B, or little b, none of that is going to be expressed. Why? Because as long as you have little c, you're going to be an albino. Once again, it doesn't matter what pigment you can produce because you lack the ability to produce pigment at all. So it doesn't matter what your B alleles are. They're going to be masked completely. This is the definition of epistasis. So we can see that any individuals that have little c are going to be white, and hence we have 4 white guinea pigs in the Punnett square below. If you have individuals that do not have little c, then that means that the color is going to be expressed. And here we've got the different black guinea pigs as well as the orange guinea pigs. So we can see in the previous slide, we know that if we have 2 heterozygous individuals, we should be getting a 9:3 to 3:1 ratio. That would be if we had simple Mendelian genetics taking place. If we have epistasis, this can mask some gene expression. And this is what we see here. When we have 2 heterozygous individuals, in this case, and if epistasis is occurring, we end up with a 9:3:4 ratio, such that 9 exhibit the dominant trait, 3 would exhibit the recessive for 1, and 4 individuals would be albino. So when you have epistasis crossing 2 heterozygous individuals, you get this characteristic 9:3:4 ratio. Here is a similar example, however, dealing with coat color in dogs. In this instance, we once again have 2 different alleles. However, if an individual has little e, that means that they will be a yellow lab, and this will mask anything that they have and anything that they're carrying in their other alleles. It's not going to be expressed. As long as you have little e, you're a yellow lab. However, if you have any other combination, either big E, little e, that will allow for the other alleles to be expressed. This will lead to the difference between black and chocolate labs. So if you have a big B, little b, you're going to be a black lab. However, if you have little b, you're going to be a chocolate lab. Once again, we get, when we're crossing 2 heterozygous individuals, which we can see at the top, we would get a skewed 9:3:4 ratio, which once again would indicate that epistasis is occurring. This slide here depicts a typical polygenic trait. Remember, a polygenic trait occurs when one trait is controlled by multiple different genes. The genes for skin color is a polygenic trait, meaning that 3 different genes contribute to overall skin color. And the combination of light versus dark alleles will determine where you fall on the gradient scale. We can see that if you have a genotype that contains mostly dark alleles, you will be on the darker end of the spectrum, and as opposed to all light alleles. And the combination of light versus dark alleles will then determine where you lie on the gradient scale. So this is very typical of other traits such as height and weight. So far we have seen that we can have simple genetics where one allele is dominant over another. We've seen that we can do Punnett squares with 1 character, 1 trait, or 2 traits. And similarly, we have seen how there can be instances where we can have more complicated genetics, such as in the case of pleiotropic genes, codominance, and incomplete dominance. To complicate things even more, environment can play a very important role on what is being expressed on an individual. This means that your phenotype might not always be strictly dependent on what your genotype is. This is one of the reasons why identical twins can have the same genetic makeup. However, they are not considered to be truly perfectly identical. There will be small, minute differences between how these genes are expressed within those individuals, even though they have the exact same genetic makeup. This means that genes can be established on a range of possible phenotypes, even with the same similar genotype. This provides the basis for nature versus nurture debate. We can see this as in the example of hydrangeas. Depending on the environment that these hydrangeas are grown in, they will exhibit a range of coloration, such as the hydrangeas that are grown in a very acidic soil with free aluminum available in the soil will have a nice bluish color, whereas hydrangeas that are grown in a basic soil will tend to be more reddish in color. The gradient between the two can also be exhibited in a range of coloration in these plants.